Today I'm going to be talking about total internal reflection microscopy of non-spherical colloidal particles. This work was done primarily by a postdoc in my group who's now moved on to be an optical engineer, Sergio Dominguez Medina, and then a PhD student in my group, Aydin Rashidi. We also worked with collaborators in Germany at the University of Bremen and also the German Aerospace Center. So one of the things that our group is interested in is the measurement of colloidal forces and measurement of dynamics of colloidal particles, in particular near boundaries. There's multiple ways that you can measure colloidal forces, three of which I'm showing here. The first two, surface force apparatus and colloidal probe AFM, are likely techniques that you're familiar with. These techniques essentially use mechanical manipulation to bring surfaces in close proximity and then measure deflections in order to infer forces. The thing that really limits you with these techniques, although they've been obviously adopted broadly by both academia and industry, one of the challenges with these is that you're limited by your ability to mechanically manipulate your arm. So for example, in AFM, the level of sensitivity with which you can measure really depends upon the ability to manufacture a cantilever that can measure such sensitive forces. What we do is we utilize total internal reflection microscopy, which is really kind of like a fundamentally different technique from these first two, in that you have a particle that's free to move. It is experiencing Brownian fluctuations. And essentially what you do is you measure these fluctuations normal to the boundary, and then utilize the observation of these fluctuations to assemble the potential energy profile, which gives you the interaction forces. The main advantage of TRM, which has been established over the last few decades, is that you're able to measure very, very weak KT scale interactions. So TRM works by using an evanescent wave, which is produced by an instant beam that's above the critical angle. So it's totally internally reflected at the interface. The evanescent wave then propagates along the surface with an intensity that decays exponentially to solution. This decay length is typically around 100 nanometers. So if a particle is within a few decay lengths of the surface, so a gap of 0 to 500 or 600 nanometers for our particular experimental setup, and that particle is capable of scattering light, then it'll scatter light with an intensity that depends very, very sensitively on the separation distance between it and the nearby boundary. This particle, because it's free to move, undergoes position fluctuations normal to the boundary. And then these position fluctuations are translated into intensity fluctuations, which are collected typically with either a photomultiplier tube or CCD camera. This would be the integrated intensity each time step of your experiment. And then these fluctuations in intensity are used to calculate fluctuations in height, which then, assuming the particle is sampling a Boltzmann distribution of heights, can be utilized to assemble the potential energy profile. So this particular potential energy profile is an expolinear curve where you have an exponential rise at lower separation distances, which is indicative of electrostatic repulsion, and then you have a linear portion at larger separation distances, which is indicative of the weight of the particle. So TRM works quite well for spherical particles, but as has become apparent in particular over the last 10 or 15 years, colloids are often anisotropic, either by nature or by design. So we're interested in a number of technologically relevant colloidal objects. One in particular that our group is kind of starting to focus on more is red blood cells. But in general, there's all types of biological colloids that are anisotropic, either in shape or in surface chemistry or optical properties. And then, of course, we know that there is such a wide range of synthetic colloids that are anisotropic. Again, I mean, this is showing mostly shape, but we know, pattern colloids can be chemically anisotropic. So there's this whole host of anisotropic particles, both in nature and in the laboratory, that really present a big challenge for experimental techniques that seek to measure interactions and dynamics of particles. Okay, so our goal here is to apply TRM to these anisotropic particle systems. And the fundamental reason that makes this a challenge is that, in the classic TRM example that I gave, I described intensity fluctuations as depending only upon separation distance, fluctuations normal to the boundary. But when you have an anisotropic particle, one may imagine that scattering will also depend upon orientation changes. And then, of course, when you assemble the potential energy landscape, you want to take into account these orientational fluctuations because the potential energy may depend upon these fluctuations in either the azimuthal angle phi or the polar angle theta. So near the beginning of this project, we just took ellipsoids that we had fabricated and put them in our TRM cell. We just looked at the scattering and found right away that scattering, if you look closely here, all of these particles are about the same height, but you can see that there's quite a bit of variation in not only the intensity of scattering, but then also the shape of scattering. So it was kind of apparent to us from the beginning that scattering intensity would not only depend upon the separation distance C, but also these azimuthal and polar angles. So a single intensity value is really not enough to obtain this information. So we began to develop a technique called scattering morphology resolved TRM. And the goal here is to deconvolute the particle orientation from the scattering intensity that is being collected. So the basic premise of SMRTIRM is that we take images of the scattering, which is actually not that different from how you would run a classic TRM experiment. In a classic TRM experiment, you just integrate the intensity. So we rather than just collecting an integrated intensity, we analyze the morphology of the scattering that is collected, which for an ellipsoid is, you can think of it as essentially an ellipse of scattering. And we fit a 2D Gaussian to this scattering morphology, which is then parameterized with something called the morphology angle and morphology aspect ratio, where the morphology angle is the scattering morphology angle, the ellipse angle with respect to the x-axis of the image. And then the morphology aspect ratio is just the aspect ratio of the ellipse that we're collecting. It's important to note, also, I didn't mention this, but for a spherical particle, the scattering morphology is nearly circular. So these values really don't have much importance for the spherical case because you have no morphology angle and you have an aspect ratio equal to 1. So then from this morphology angle and morphology aspect ratio, we hope to determine the azimuthal angle and then also the polar angle, and then determine the height independent of these orientation measurements. So really the main difference here between SMRTIRM and conventional TRM is that the scattering image is analyzed simultaneously with the scattering intensity. So we're actually interpreting the morphology of scattering that we're seeing. And the hypothesis driving this is that this scattering morphology encodes information about particle orientation. So we make ellipsoids in the lab. We do it with a somewhat well-known technique at this point, where you disperse polystyrene beads into a polyvinyl alcohol film. You raise the temperature of the film to above the glass transition temperature. You stretch uniaxially. This is a picture actually of a biaxial stretcher that we use at the University of Akron. You can stretch actually biaxially if you'd like and make more complex shapes. But for this talk, I'm just going to be talking about ellipsoids, so prolate ellipsoids. So we uniaxially stretch, and we can stretch to different ratios and get low aspect ellipsoids or high aspect ratio ellipsoids. So the way that the experiment is conducted is that we suspend the ellipsoids into solution with a large amount of salt. The salt then screens electrostatic repulsion between the ellipsoids and the nearby boundary. And then because of the screening, the particles deposit at randomly different azimuthal angles. And then we illuminate with an evanescent field such that we get a bunch of particles at different azimuthal angles with scattering that is then analyzed as I described. Okay, so here are some images from our experiment on the left-hand side. We have panels with bright field for low aspect ratio ellipsoids and panels with bright field for high aspect ratio ellipsoids. And then we also have the images with just the scattering from the evanescent wave for low aspect ratio ellipsoids and high aspect ratio ellipsoids. The azimuthal angle determined from bright field is given in the bright field images, and then the scattering morphology angle is given in the evanescent wave images. And we can see right away that there's a correlation between the two. As you systematically change the azimuthal angle, you can systematically change the morphology angle. And we did this experiment many dozens and dozens of times for low aspect ratio and high aspect ratio ellipsoids, and we find that there's this correlation between the two. But essentially, we utilize the T-matrix method to simulate the scattering field, and then with knowledge of the scattering field and knowledge of the optics of our collection system, our particular collection system for our experiment, we can produce images that would be analogous to the images that we collect in our experiment with our CCD panel. Okay, so these are simulated images. Again, we have bright field on the left-hand side with the angle as determined from bright field. And then we have for both low and high aspect ratio ellipsoids, and then we have the evanescent wave scattering images, again, simulated. And then along with those, we have the morphology angle as determined from analysis of these. So up in the right-hand corner, we have the simulated data with the experimental data for both low and high aspect ratio ellipsoids. And you can see indeed that it's nonlinear, that there is a bit of curvature in how the scattered morphology depends upon the azimuthal angle for both cases. What's nice about the simulation tool is that we can have much lower sensitivity to picking up scattering. Again, I'll mention this near the end, but so we can pick up these kind of blind regions that we didn't see in the experiment in the simulation. So it angles lower than 60 degrees and higher than about 120 degrees for the high aspect ratio ellipsoids. So the average error in determining the azimuthal angle for these data was about 2 degrees, which is important to note is equal to or smaller than the angular resolution for bright field observation in the azimuthal angle. And it's important to realize that the that the azimuthal angle is actually the easier of the two angles to determine in bright field microscopy. The polar angle is actually quite a bit more difficult to determine accurately in bright field microscopy. So we wanted to determine what impact the scattering would, or what impact the polar angle would have on the scattering. So we wanted to determine what impact the polar angle had on the morphology. And first, what I want to show you is the data in the upper right-hand corner here, which is the morphology angle. So this is the parameter that I've been talking about for most of my slides thus far as a function of azimuthal angle for two different polar angles for both the low aspect ratio and the high aspect ratio ellipsoids. And indeed, we find that there's very little change in the morphology angle. So what that means is that the morphology parameter, the angular parameter, does not change sensitively as a function of polar angle. However, if we look at the morphology aspect ratio, so these simulated images are of evanescent wave scattering for two different polar angles and different azimuthal angles, we see that, okay, at 0 degrees, there's really no change in the aspect ratio. But actually, as we change azimuthal angle, there is a sensitive change in the aspect ratio as a consequence of changes in polar angle. Okay, so the images show that. And then if we generate more simulated images over a wider range of azimuthal angles, those data are shown here, where the morphology aspect ratio is shown on the y-axis as a function of the polar angle. And we can see immediately that there's really extreme sensitivity of the morphology aspect ratio in response to changes in really small, actually small changes of the polar angle. So 0, -10, and 10 degrees. So it's a range of 20 degrees in the polar angle. And we're seeing very large changes in this morphology aspect ratio as a function of the azimuthal angle. So what this means is that this is really a promising development for SMRTIRM because mainly we can determine the azimuthal angle from the morphology angle and then independently determine the polar angle from the aspect ratio. So we really have two different parameters here that can be used to independently find these two different orientations. Okay, so in conclusion, I've described how SMRTIRM combines scattering intensity and image analysis to decode the particle position and orientation. The morphology angle and the morphology aspect ratio are really the recorders of the azimuthal angle and polar angle, respectively. Variations in the azimuthal angle can decrease scattering intensity to almost 0 at constant height, while small deviations in the polar orientation also can cause large changes in intensity. So really, what we've done here is we've set up the machinery such that by combining the morphology angle and morphology aspect ratio, we can achieve 3D positional tracking and orientational tracking of a colloidal ellipsoid. Okay, and then I'll just thank the individuals who are responsible for the work. So Aidan Rashidi and Sergio Dominguez Medina were really the drivers of this work. Gary Yan also assisted us, and then our collaborators in Germany were extremely helpful with the simulation. Thank you.